Continuing the chapter on classification. I don't know how many people are still familiar with the Brady Bunch, but that's what this is, and that's the way we became the bacteria bunch. And on the bottom it says single cell sitcoms. And those are supposed to be bacteria, which used to be a kingdom. Now it's two kingdoms inside a particular domain, or it's two domains that are separate from one another. See how disagreement works? And something I read within the last 2 months, they may be taking one of those domains and adding it to ours. They're finding, looking at genetics, that maybe it's much more closely related to our group and it shouldn't be as separate. And this is how these arguments get going. So you'll notice this is a fairly old overhead. You'll notice even this is confusing. Is that it's got a combined group there divided into two domains, and then a bunch of groups there under one domain. What we're going to do is we're going to work this as a six-kingdom system, just because that's the way it's going to make sense. As I said, the group names are going to be similar no matter whether you call them a kingdom or a domain or what you call them. Although this has now been changed to that. But the names are going to be more or less the same. Because there's so much disagreement, there's actually a lot of different kingdom systems. When I teach animal biology, every time they put out a new edition of the book, they've changed the kingdom system. They've taken some groups and either raised them to the level of kingdom or combined them and put them inside kingdoms that they didn't used to share. Again, moving stuff up and down the ladder. They don't change the names of the groups, so I still know which groups are which. Here, this is kind of a classic introductory biology approach. When I learned biology and first started teaching it, there was a 5-kingdom system. They've split one of the kingdoms because they've learned some stuff about it that basically amounts to, no, these are too different to leave them in one group. The types of cells are different. There are two kingdoms with what used to be called bacterial cells, little bitty cells that are simpler inside. They don't have the contained chambers separate from the surface, and they don't even have a chamber for their DNA. They don't have a nucleus. But there's all sorts of other chambers that happen in cells that they don't have as well. There are two different prokaryote kingdoms. The eukaryotes, here's the eukaryote cell, here's the little prokaryote cell, big difference in size. And the eukaryotes have a bunch of internal chambers, including a special chamber for DNA processing, that's the nucleus. We're not gonna really get much into the bits and pieces of cells. I do that in my other classes. Here, we're just gonna talk kind of in simple terms about the types of cells. Now, sometimes I need to lie to you. I will tell you when I'm lying to you, but then I'll tell you why. But sometimes I need to lie to you because, well, here's a reason. The differences between these are molecular based, they're molecular biology based. So I don't want to get into that. I'm going to give you a set of differences that isn't 100% reliable, but it's a pretty good way to grasp the differences between the two groups. And so it's what we're going to use. It's not exactly dividing between the two groups, but it works okay. there are environments in the world that are pretty extreme. They're very hot, or they're very cold, or they're acidic, or they're dry, or they're salty in certain ways. Those are extreme environments. And these, which all used to be called bacteria, when you're looking at extreme environments like hot springs or very salty lakes or acidic lakes, the bacteria you find are more likely to be in the archaea group. Bacteria out here in the real world, in the regular world, much more likely to be Monarans. Remember I said it's not strictly a good division? There are archaea that live out and around us. There are some Monaras that live in extreme environments. But your best guess, if you find bacteria in an extreme environment, like you're pulling up a drill head from 3 miles down in the bedrock and you find bacteria on them, really good chance they're going to be archaea. It's a pretty extreme environment. Whereas if you're just scraping bacteria off of a surface around here and you're going to culture it and figure out what it is in the lab, they're very likely to be Monera. The real differences are they process their DNA somewhat differently, they build their casings differently. There are molecular differences between them, but we don't want to get into that because we need to get into the molecular stuff to get into that. For the eukaryotes, we've got the protista, when I first learned this, they were always single-celled eukaryotes. And the simple algae, the algae that have always been in the oceans that are not descended from land plants, were part of the plants. And at some point, the plant people kicked them out. Said, here, protista people, you got the algae now. And they went, yeah, but we used to teach that it was only single-celled, and these guys are sheets of cells. And they went, we don't care. Is that we're not gonna include them in our group anymore. It gets weird. So the protista are single-celled but more complex cells like our cells, and some what they call marine algae. Everything else is multicelled. Multicelled, photosynthetic, have adapted to living on land even if they've gone back into the water. Those are the plantae, the plants. Is that photosynthesis isn't enough because there are photosynthetic protists, there are photosynthetic archaea, there are photosynthetic Monera. Being able to do photosynthesis isn't particular to any one kingdom. There are some kingdoms that don't do it, like the next two, but all of these other ones you can find groups that do photosynthesis. But you don't find multi-celled photosynthetic land plants in any other group. In the animals, Animalia. There's something about writing on overhead, so it's real easy to leave letters out of words. Why is he writing this weird thing? Animals move. Well, unfortunately, not all the animals have gotten that memo. And so animals do usually move. I did put it down at the end, but there are a lot of exceptions. There are sponges don't move, corals don't move. There's a lot of animals that what are called sessile. They sit in one place for the whole lifetime. And it's weird. In order to set the animals apart from the other groups, we need to talk about their digestion and absorption. Because weirdly enough, we are very much more closely related to the fungus group than to any of these other kingdoms. And one of the things that makes us different is how we get our nutrition. The fungi are considered multi-celled. There are a few single-celled types, like yeasts. And they digest and absorb across their outer surface. Okay. Again, I have to do a usually here, because it isn't always true. But a typical fungus usually is a network of strings of cells called hyphae. You don't need to know that. And so a mushroom is actually a whole bunch of those fibers collected together in order to produce and release spores. The organism itself, the mushroom fungus, is actually very thin fibers spread through the soil. That's why if you pluck the mushroom off, it doesn't seem to be connected to anything. It doesn't have roots, but it is connected to fibers, but they're microscopic. Is that if you had, the right sort of imaging system, you could see them in the soil. But the only time they gather them all together into a structure that needs to stand up in the air is when they make mushrooms. Is that there are some funguses that are not always just a network of fibers, but that's typical for funguses. They tend to extend. A spore will generate the fibers and they spread out, and they secrete chemicals to help them break down food materials, and then they absorb it across the outside of the fibers. We've got internal chambers for digestion and absorption. They do it across their outside surface. It's a major difference between the two. Chemically, there are other differences, but again, we don't want to get into that. So those are our 6 kingdoms. There's an idea of the funguses. Sometimes if you see old dead wood, is that you'll see what looks like spider webs across the spaces. And those are type of fibers, fungus. Not a single cell strand, it's actually multi-celled in order, otherwise it wouldn't be strong enough to make the reach. But again, made up of fibers. Okay, there's a couple more things to cover. I'm going to write it and then I'm going to describe them, so I'm not going to always write down the description here. The classic approach to classification is similar to what we do with people. How closely related am I to somebody? Well, do we share a father? Do we share a grandfather? Do we share a great-great-grandfather? Do we share a great-great-great-grandfather? Is it how closely related we are depends on how many generations we have to go back before we share the same relative. Systematics takes the same approach. It builds trees, and at every tree branch, there's a group that's an ancestor of both groups on the branches. And so it's built on shared ancestors. Cladistics builds trees and then looks at features that make one group new and different from the other groups. And so the splits on a cladistics tree show where in the past there's a new feature that's characteristic of this group but not of the other group that keeps going on in another direction. And genetics, which is, they haven't really worked all of the rules out for, looks at similarities, homologies, structural similarities in genes and DNA sequences, and how closely we are related to other things. We share similar genes, right? That's what they're doing with the viruses now. They're looking at the genetic patterns to see, this is a new strain, that's not a new strain, that sort of thing. This is what they call a cladogram, where the split here is you're coming up with a backbone, a vertebral column. This split produces hair and mammary glands. Here you've got the reptiles. They didn't have that. They split off. Then these guys lay eggs. The split here is for gestation, the development inside the body. Then you've got the marsupials, the ones with the pouches. Their fetuses leave the womb pretty early and get into the pouch to develop. These guys stay inside the mother to develop, so that's a long gestation. So these are features, and that's shown by the splits. the smallest groups. How do we decide what belongs just in a species? Early on, it was like, I know it when I see it. They just look too different. Is that foxes and dogs just look different enough, they're different species. Well, are gray foxes and red foxes different enough to be different species? Yeah. So it came down to that. As people started to collect animals and put them together and find that they get bored and they have sex, they found that some animals that look very similar to one another can't make babies together, like donkeys and horses. Well, no, donkeys and horses can make mules. That was another thing they discovered. Mules can't make more mules, though. Yeah, is that you can combine them and get babies, but the babies can't make babies. So they just fiddled with the rules and fiddled with the rules and fiddled with the rules. None of the rules fit every single circumstance. It's biology. We're a science of exceptions. Almost any rule you come up with, if you keep looking, you're going to find exceptions to the rule. And right now, probably the best species definition we have right now, so get ready to write this down, is a lot of times we look at a species as a group that in nature just reproduces within its particular group. It's kind of a way of saying, let's let them show us who belongs in a group and who belongs in another group. Is that they won't, even though you've got two things in the same area that look very similar to one another, they know we reproduce just within this group over here, we reproduce just within this group over here. And that's a basis for making species. How you use genetics to determine that, how different do the genetics have to be in order for you to split a species off or a subspecies? They're now working the levels out for that. A lot of disagreement. Welcome to science. It's in the next chapter, we'll talk about how it's a product of people, and people like to disagree about things. They like to define things their way and not necessarily somebody else's way. But that's it for the classification chapter.